Hello, my name is Bridget Blaszak, and today I will be presenting my project, which is titled The Effect of Granular Stabilized Chlorine on the Presence of Regeneration in Brown Planaria. The independent variable in my experiment was the concentration of granular stabilized chlorine, a commonly used pool chemical, in a spring water solution. Granular stabilized chlorine consists of hypochlorous acid and acetic acid. The dependent variable in my experiment was the presence of regeneration in the form of a blastema in brown planaria. Brown planaria are freshwater flatworms that have a distinct regenerative ability, and therefore they are commonly used in experiments concerning wound regeneration. The rationale for my project includes the common myth that exposure to chlorine will accelerate the wound healing process, and yet there are no scientific studies available that investigate this common belief. And the purpose of this experiment is to explore the effects of granular stabilized chlorine on the presence of regeneration in brown planaria, and the results of this experiment may be applied to future studies concerning the effects of pool sanitation products on human skin wounds. Why this project? Personally, I've been involved in the sport of swimming, and people frequently mention that chlorine heals wounds, and yet when I scientifically investigate this, I cannot seem to find any significant scientific studies that either support or do not support this belief. And my key question is, how does granular stabilized chlorine affect the wound healing process? The real-world application for my project concerns chronic wounds. Chronic wounds are an emerging issue in the medical field as they have a prolonged healing process and are often caused by common underlying conditions such as atopic dermatitis or diabetes. And current treatment options like topical steroids have adverse long-term side effects. Chlorine byproducts may emerge as a prospective treatment option for chronic wounds. Some resources that I found helpful in the completion of my project included access to a fume hood in order to safely mix chemicals, the ability to confer with an experienced person in the field of science, such as Miss Denise Williams, and certain pieces of glassware like pipettes in order to efficiently measure out and disperse chlorine solutions in my project. My hypothesis states that if brown planaria are exposed to a higher concentration of granular stabilized chlorine, then they will all exhibit regeneration after 14 days of exposure. Here's my experimental design diagram, and something I like to point out is my levels of independent variable, and I would like to justify them. My first level, which contains no granular stabilized chlorine, is my control. My second level, which has 2.5 milligrams of chlorine, simulates the amount of chlorine that's used in backyard pools. And my third IV level, which contains 4.0 milligrams of chlorine, simulates the amount of chlorine that is most typically used in municipal and public pools. Within my procedure, I performed my experiment in the Godwin High School lab underneath the supervision of Ms. Williams. And in order to perform this experiment safely and effectively, all chemicals were mixed underneath a fume hood. An apron, goggles, a mask, and nitrile gloves were always worn when directly handling granular stabilized chlorine, and the brown planaria were observed underneath a compound light microscope to ensure that accurate classifications were made. In the beginning of my procedure, I created 3 granular stabilized chlorine solutions consisting of spring water and granular stabilized chlorine under a fume hood. Then I measured out 10 milliliters of each solution with a glass pipette and placed them into their own individual petri dishes. I then repeated this 25 times, so I had 25 petri dishes for each IV level. And then I bisected 75 brown planaria underneath a compound light microscope and left these specimens underneath a stable conditions in a stable environment in the petri dishes for 7 days. Once 7 days had passed, I fed the planaria 50 milligrams of hard-boiled egg, and then after 14 days, the specimens were observed underneath a microscope and, with the help of a chart from a pre-existing study, were classified as blastema present, no blastema present, or death. At the conclusion of my experiment, the planaria were frozen and thrown away, and the remaining chlorine solutions were then diluted and poured down a drain. Some constraints I encountered in experimental design development included the inconsistent distribution of chlorine among trials, as mixing the solution for a set period of time would help me rectify this in future experiments, and I had some difficulty bisecting the planaria below the pharynx as they were very quickly moving. I can use chilled water in the future to help slow down this movement so I can exactly bisect the planaria just below their pharynx. Here are some images from my procedure. This is the creation of the granular stabilized chlorine solutions and their measuring. This image depicts the bisection of a planaria, and here is a planaria right after bisection. Here's the procedural diagram I used to sort of determine what was a blastema and what was not. Anything in the bottom row, so figures 3 through 7, were considered a blastema. Here, moving on to my results, is my graph, and something most striking about this is, as you see, in my highest IV level, all of the planarian exhibited death. Here is my statistical analysis, and the most striking part about my statistical analysis is that 100% of my data was statistically significant by way of a chi-square. The results of my experiment in the frequency distribution of my control showed that about half the planarian exhibited a blastema, but then the frequency distribution of my highest IV level shows the lethal effect of the concentration on the planaria, as all the flatworms were classified as death, and as previously stated, all my data was statistically significant at a level of 0.01. The data did not support my research hypothesis, but rather contradicted it, and it shows that there's an inverse relationship between that concentration of granular stabilized chlorine and the presence of regeneration, as when you compare the modes of the control and the 2.5 milligram concentrations, which are the blastema present and no blastema present and present, it shows that the amount of planaria that were exhibiting a blastema is decreasing, and a similar effect can be observed for the modes of my 2.5 milligram IV levels and my 4.0 milligram IV level. Some strengths and limitations of my data, and some strengths include that resources from a previous study were used in data collection, that procedural diagram that I previously showed, and then the specimens were examined under a microscope, which allowing for more accurate classifications to be made. Some limitations include that qualitative data introduces the error of human judgment, and I use a less complex invertebrate model that is not as similar to mammals as, for example, mice. Here are some images from my results, which include here's a planaria that did not exhibit a blastema. This planaria was classified as death, and this planaria did exhibit a blastema. Moving on to my conclusion, the results of this experiment did not support the research hypothesis. In fact, the data suggest an inverse relationship between granular stabilized chlorine and the presence of regeneration in brown planaria. The rapid decline in the presence of regeneration with increased chlorine concentration can be explained by previous studies, including a 2019 study that found that calcium chloride impairs the organism's regenerative ability. Calcium chloride is a chlorine byproduct that is very similar to granular stabilized chlorine. My scientific reason for the results comes in the nature of the blastema's tissue. For natural normal cell differentiation to occur, direct contact must occur between the epithelial cells and the parenchyma. The parenchyma is the tissue found between an invertebrate's body wall and the organs. And a study involving magnesium chloride found that chlorine interferes with the direct contact, and then normal generation will not occur. Some unexpected difficulties I encountered, for example, this problem in the inability to accurately and quantitatively measure the change in length of planarian after the regenerative period, as the length of a blastema is quite marginal and it's very difficult to measure with a ruler as planaria are quickly moving, and my solution was to switch from a quantitative to qualitative data model in which the planaria were categorized based on a chart from a previous study. Some sources of error in my experiment include the unequal distribution of granular stabilized chlorine within the solution, as the chlorine solutions in future experiments will be mixed for 1 minute in order to ensure that the chemical is evenly distributed, and then my switch from quantitative to qualitative data. And now I will take photos of the planaria before and after the regenerative period to show exactly how much changes have been exhibited, and then the varying amounts of solution within petri dishes caused by air bubbles in the glass pipette. And in future experiments, the experimenter will verify that no air bubbles are present in the glass pipette. My recommendations for future studies include a more complex marine model that will make the experiment more applicable to humans, and the solutions now will have to be administered through a topical gel. And another future study may compare the effects of different forms of granular stabilized chlorine on the presence of wound regeneration in brown planaria, as previous studies involving hypochlorous acid and chlorine dioxide showed a direct relationship between chlorine concentration and wound regeneration. Some important takeaways from my research include that granular stabilized chlorine appears to impair the regenerative processes in brown planaria, and it's deadly when given in higher concentrations. And previous studies suggest that other chlorine byproducts have a positive effect on wound regeneration, and future studies may compare the effects of different chlorine byproducts on wound healing. Here are my literature cited. I use both peer-reviewed and non-peer-reviewed sources. And for my acknowledgments, I would like to thank Ms. Samantha Cope, Ms. Denise Williams, and my parents for all their help in the completion of this research project. Thank you.